This program is brought to you by Emory University.
Good morning, everyone. Welcome to Cardiovascular Grand Rounds. This morning we have a special treat. We have Dr. Thomas Wang visiting us from Vanderbilt. Those of you who don't know Dr. Wang, he was sort of Harvard all the way, Harvard College, Harvard Medical School, did his internship and residency at the Mass General and his fellowship, and stayed on there as faculty through 2013, at which point he was recruited to Vanderbilt to be their chief of cardiology. Dr. Wang's career is a very exciting one, major contributions in a large variety of areas, focusing a lot on a series of biomarkers and use in large population studies, particular emphasis on this natriuretic peptides that he's going to talk to us today. He has served on numerous committees, standing study sections, NIH, editorial board of circulation, the research committee for National Research Committee for the American Heart Association, etc. And we're really pleased to have you here today. Dr. Wang, welcome.
Great, thanks so much, Bob. It's a real pleasure to be here today. It's always a pleasure to visit other cardiology divisions, but a particularly nice opportunity to come and visit a neighbor to the south of us. Many of our faculty spent significant amounts of time here in training or medical school, so I feel like I know Emory a bit already. So as Bob mentioned, I have had a long-standing interest in biomarkers since my training. Clinically, I also specialize in heart failure, and so it would be natural that I would be interested in the natriuretic peptides. BNP as a biomarker for heart failure was becoming popular around the time I was finishing training. This is from Ellen Maisel's randomized trial in the New England Journal in 2002, showing what was known then, but demonstrating in the setting of a prospective study that individuals who come in with heart failure have dramatically higher levels of BNP than individuals who do not have heart failure. If you actually look at the scale here, typically around 500 or 600 for mature BNP compared to healthy or sort of non-heart failure individuals in the same study, which are levels several orders of magnitude lower. So the purpose of the talk today is not so much to revisit what many of you already know about the utility of BNP in the clinical setting, especially for heart failure, but to hopefully get you to think a little bit differently or more broadly about the role of the natriuretic peptides in health. And for this, I'm going to go back, as is the custom for outside speakers, to work that was originally done at the institution that I'm at now. And so Earl Sutherland was one of the fathers of cardiovascular pharmacology, not just at Vanderbilt, but obviously all around. He won a Nobel Prize in 1971 when he was on the faculty at Vanderbilt for the discoveries that led to the concept of second messengers, particular work surrounding cyclic AMP as a second messenger for hormonal signals. In subsequent decades at Vanderbilt, a number of investigators, including Jackie Corbin, have had an interest in cyclic GMP. And the reason, of course, that that's relevant is that cyclic GMP is the intracellular second messenger for two major systems, the natriuretic peptide system and the nitric oxide system. And for the natriuretic peptides is really responsible for the vast majority, if not all, of the downstream actions, including smooth muscle relaxation. And so, as you all know, the natriuretic peptides in the body serve as sort of an endogenous inhibitor of the renin-angiotensin system. And so, whereas the renin-angiotensin system has a number of actions, including sodium retention and vasoconstriction, the natriuretic peptides do basically the opposite. They help you excrete salt, they vasodilate, and they're also anti-hypertrophic hormones. And so one might consider that in the context of heart failure that one might actually have too little of ANP and BNP because heart failure is a salt-retaining state. It was actually work dating back to the mid-'80s by John Burnett and his colleagues at the Mayo Clinic that really helped disseminate the concept that the natriuretic peptides were in fact elevated in the context of heart failure. As John wrote here, congestive heart failure reflects not an ANP deficiency state, but rather in a compensatory increase in peptide release. And further on, that he noted in the small number of human subjects that there were very high circulating ANP levels despite the existence of sodium retention and edema formation. And of course, the conventional model then is that the heart, when under cardiac overload, secretes high amounts of ANP and BNP into the circulation. These peptides have their downstream effects, the salt excretion and the vasodilation, inhibition of the RAS system. And those effects collectively serve a compensatory role to offset the initial increase in cardiac overload, although like all compensatory actions, they don't fully offset those initial effects. And so over the years, a number of clinical studies, starting with Allen's study and others like it, led to the idea that in heart failure, you typically get mature BNP levels in the hundreds or often well over 1,000. We did a number of studies in Framingham and other cohorts, and others like Maggie Redfield and John Burnett at the Mayo did similar studies looking at BNP values in healthy individuals, typically showing that in a healthy middle-aged individual, the BNP level that you'd find, if you can measure it, is around 5 to 15 picograms per ml. And I say if you can measure it because often they're even undetectable. They're very low levels of BNP. And so there's an enormous spread between healthy individuals and individuals with heart failure, which again has led to its utility as a biomarker for states like heart failure. So I just wanted to then move on to some clinical vignettes to hopefully illustrate most of what I'm going to talk about today. These are actually taken from the Vanderbilt electronic medical record. As some of you know, Vanderbilt's EMR, almost in its entirety, is mirrored in a research database called the synthetic derivative. And so the faculty members shown below, including Quinn Wells and Deepak Gupta, have helped collect cases like this, which I'll show right now. There are two cases. One is of a 59-year-old woman with ischemic cardiomyopathy, EF of 25%. She is hypertensive and obese. And at left heart cath at Vanderbilt, she has an LV EDP of 32 millimeters of mercury. And so I think no one would quarrel with the fact that this is a woman who has overt systolic heart failure and decompensated hemodynamics. And yet her BNP was only 12 picograms per ml. In fact, if you look throughout her electronic medical record during this period, she has consistently very low levels of BNP. Patient number 2 is a 69-year-old African-American woman with heart failure with preserved EF and shortness of breath. She too is obese and hypertensive, and at her cath she has a wedge of 27. Echo shows very thick walls consistent with her diagnosis of HFpEF. And she has levels of BNP of 29 picograms per ml, and again, consistently below 30 throughout the course of her hospitalization and surrounding hospitalizations. And so these cases are meant to illustrate that sometimes in the context of heart failure, you get BNPs that are just surprisingly low. And these are patients for whom there really is no clinical question that their heart is overloaded and that they should be making lots and lots of BNP. And so what I want to address during this talk are a couple questions. One, are there actually states of natriuretic peptide deficiency where people do not make as much BNP or ANP as you would expect them to make? And what would the consequences of that be? Secondly, supposing that that does exist, who gets natriuretic peptide deficiency? And then lastly, can this deficiency state be reversed? And so the reason to even think about a deficiency state is, of course, the natriuretic peptides are hormones, and like any other hormonal system, there is reason to suspect that there may be deficiency or excess states. If you think about the thyroid hormone, PTH, insulin is a hormone, there are clearly states of deficiency and states of excess. And for endocrine conditions, these states are really relatively well defined. The hormones that the heart makes, the natriuretic peptides, may then be associated with these states, but we know, in fact, nothing about them or whether, in fact, they exist at all. So the first question would be to ask, is there some sort of genetic predilection to making too much or too little natriuretic peptides? This is from a study that was done right as I was leaving fellowship, again using the Framingham cohort, to look at the heritability of BNP and ANP. This is being done by looking at related individuals and comparing their levels, and then unrelated individuals like husband and wife pairs and looking at their levels. And in fact, what you find is that there's a surprising amount of heritability for both of these peptides. Heritability being defined as the amount of inter-individual variation in a trait, in this case natriuretic peptide levels, it seems to be attributable to genetic factors. So about 40% of the circulating levels in a healthy individual seems to be attributable to genetic factors. And so what are these specific genetic factors? So this is from a GWAS of natriuretic peptides now in about 16,000 people across many NIH cohorts. And this was done in collaboration with the consortium, as well as Chris Newton-Che, a former colleague, and Dan Levy up at Framingham. And many of you have seen these Manhattan plots. These peaks represent areas in along the chromosomes that have the highest associations with the trait. And as you can see, there's a dramatically high peak right at the end of chromosome 1, which in fact overlies the genes, or the pair of genes that encodes AMP and BNP. So not terribly surprising. Just of interest, although I won't talk about them, there are a couple of other peaks away from that locus that also seem to be associated with natriuretic peptide levels. So about this peak, the best SNP at this peak is a SNP associated with ANP levels called RS5068 with a very low p-value, as you can see there. We have found in not just Framingham, but a number of other cohorts of white individuals that the allele of RS5068 that one has is somewhat deterministic of whether one has high levels or low levels of AMP. And so this from a Finnish cohort, the G allele, which is the less common allele, the minor allele, is associated with higher levels of AMP, whereas the A allele is associated with lower levels. And there's about a 15 to 20% difference from the highest to lowest level according to genotype category. So a relatively significant and substantial effect. But just to get an idea of how much of an effect this was, we did a series of physiologic studies. These were led by one of our postdocs, Pankaj Arora, who's a faculty member at UAB currently. There are 30 individuals that we took that were selected on the basis of RS5068 genotype. This top dash curve shows the individuals who had one copy of the G allele, so they are heterozygous at this locus. And the bottom curve are those with no copies of the G allele. Just to give you an idea, about 90% of white individuals would fall in this AA group, the bottom curve, about 10% of individuals in the top curve. And so what was done is the individuals were put on a week of a high salt diet to sort of put them on a common background, and then brought into the GCRC and given 2 to 3 liters of normal saline between 0 and 2 hours, and then followed for 8 hours. And as you can see, both groups, as expected, when you load them with saline, have an increase in their ANP levels, about 50 to 60%. But clearly, the G allele group, the individuals who carry a copy of the minor allele, have a much brisker increase and have a higher set point of ANP levels achieved throughout the infusion in the 8 hours afterwards. In fact, if you look at the magnitude of the increase, the difference between these curves is comparable to the excursion that you get on any individual curve during the 3-liter infusion. So one way to think about it intuitively is that the effect of having one copy of the G allele is about the same as the effect of getting 3 liters of normal saline infused through your IV, which again, in that context, suggests that it's a relatively significant effect. We didn't have too many individuals that we came across who had 2 copies of the minor allele, the homozygotes, but we did have 2 shown in red, and as expected, they have a dramatic increase in their excretion of ANP during the course of the saline infusion and afterwards. And so the question would be, if we accept that there are genetic factors that predispose to whether you make more or less ANP, what are the consequences of this? Of course, the traditional way of thinking about this is to look at what happens in a mouse when you either get rid of ANP or make it make more ANP or BNP than it normally does. And the phenotypes that you get in the mice are pretty predictable based on the physiologic actions of these peptides. A mouse that does not have an ANP gene or a BNP gene gets, in the case of ANP, salt-sensitive hypertension and LVH, and in the case of the BNP knockout, gets a prominent cardiac fibrosis. The flip side is that mice that make too much BNP tend to have lower blood pressure. And so we can ask this question in humans now that we have large data sets in which people have been genotyped. And so again, if you look at this RS506 finding with the G allele, the one that gives you higher levels of ANP, you find that if you look at very large cohorts, we initially did this in the Framingham study in a Swedish cohort in Malmo, and then in subsequent larger studies like Global BBGen and the International Consortium of Blood Pressure Genetics, that there's a highly significant association between having this allele, the G allele, and lower risk of hypertension, as you would predict based on the physiology. And similarly, in another consortium, the CHARGE consortium, the G allele was associated with lower LV mass, again, as you would predict based on the animal data. The other question is whether there are other consequences of making too little or too much ANP or BNP other than the expected differences in blood pressure and LV mass. One of the interesting phenotypes that's come out in the last 5 years in these transgenic mice for BNP are they also seem to be protected against weight gain. And so these mice that make more BNP gain less weight on a high-fat diet, and they seem to be protected against glucose intolerance. And so going again to the humans, these are data from John Burnett's group, who has looked now in the Rochester cohort as well as cohorts on the Mediterranean, showing that individuals who carry 1 or 2 copies of the G allele have a lower prevalence of metabolic syndrome and a lower prevalence of obesity, again suggesting that in humans, as in the animals, there's some protective effect of this genetic ANP excess. There are also some older data from a diabetes consortium. This is about 60,000 individuals in a case control study showing that if you have an allele associated with higher BNP levels, you have about a 15% lower risk of diabetes. So again, supporting the overall hypothesis. These are the mice data that I mentioned earlier. These are the mice that overexpress BNP, and so as you can see, after being on a high-fat diet, they have improved glucose tolerance. And as shown here on the high-fat diet, the overexpressing mice have less fat accumulation after the end of 3 weeks. These are actually coronal cuts through the mice showing that on the high-fat diet, the reduced accumulation of fat is actually particularly prominent around the viscera. So it's less visceral adiposity, not so much less subcutaneous adiposity. This observation led us to look again back in the Framingham cohort where we could compare circulating BNP values and subcutaneous and visceral adiposity measured using CT scan in about 2,000 individuals. And again, as in the animals, there's a nice inverse relationship between amounts of visceral fat and BNP values, which you do not see in the subcutaneous fat. And so I won't go into the mechanisms for the protective metabolic effects, but there have been a number of experimental studies done in the last 5 or 6 years to really try to understand this. There are really probably at least 2 things going on. One is that stimulation of cyclic GMP through this pathway seems to promote lipolysis, both in animals and in humans, shown in this leg. And the other interesting finding is that animals who have increased activity in the system also have increased mitochondrial biogenesis, and so they have more active fat and an upregulation of their brown fat. And so this is just one slide from those studies. This was done by Sheila Collins' group at the Sanford Burnham Institute, showing that these are mice that do not have the clearance receptor for ANP and BNP, so they have higher circulating ANP and BNP values. They tend to have smaller fat cells, so more mature fat cells that are less lipid-filled, and they have upregulation of genes associated with brown fat. So again, they seem to have this increased thermogenic program suggestive of increased energy expenditure. So I'm just going to switch then and talk for a couple minutes on this idea of who might get natriuretic peptide deficiency and what would the possible mechanisms be if we accept that it exists. And so again, just to revisit the overall model here, we've seen that both humans and animals that seem to make more ANP and BNP have lower blood pressure, lower LV mass, and a protective metabolic phenotype. Conversely, you would expect then that individuals that have a handicap in their ability to make natriuretic peptides would have hypertension, LVH, and obesity or diabetes. And so again, the question is what might cause this? The first thing that I want to talk briefly about and revisit is what are the genetic mechanisms that might cause this. And for this, I'm going to go back again to that finding with RS5068. So again, this was work done by Punkage and colleagues when I was back in Boston. RS5068, like many common variants that are associated with disease, is in the non-coding region of the ANP gene. It's in the 3' UTR of the gene, again shown there with the A allele, which is the common allele, or the G allele, the less common allele. The 3' UTR is a common target of microRNAs, which led Punkage to hypothesize that maybe this involved disruption of a microRNA relationship. And so he went back to the databases and found 3 microRNAs that had seed sequences that were predicted to overlap the 3' UTR, the ANP gene, and did experiments with those. And so the hypothesis here is that in the common state, in the 90% of white individuals who have the A allele, there's a specific microRNA, and it turns out that microRNA 425 is the one that seems to have this characteristic that binds to the 3' UTR of the ANP gene. And like all microRNA, that causes a inhibition of transcription or translation of the message to protein. And so the microRNA is a natural inhibitor of ANP production. Individuals with a G allele, because they can't bind the microRNA, then have a release of this inhibition, and so that's why they make more ANP. And so the experiments that Punkage did were initially with this luciferase reporter system that was attached to the 3' UTR from that either featured the major allele or the minor allele. And what Punkage showed was that, as hypothesized, in the major allele condition, in the common condition, transfection with the microRNA resulted in reduced ANP production, whereas in the minor allele condition, there was no consequent reduction. Punkage then went on and did some clever experiments to show that this base pair change was sufficient to cause the effect. And so what he did is that if the state that's happening now for the G allele is blocking the binding of the microRNA, he hypothesized, well, if you just changed not the G allele, kept the G allele, but if you changed the base pair in the microRNA that you predicted was going to bind to the G allele to something that was complementary to the G allele, in that case, this modified microRNA should then bind the minor allele construct, and then you get the expected reduction in ANP production. And so, again, these are experiments now with the modified microRNA predicted to bind to the minor allele. And as shown here, you can now then recapitulate the finding of reduced ANP production with the, now with the minor allele, because you're using the genetically engineered microRNA. These are, I won't show all the experiments, these are some further experiments now in human cardiomyocytes showing that direct transfection of microRNA results in reduced expression of ANP, as well as a reduction in the levels of ANP found in the media. And it's actually quite a significant reduction that you find. Just to close the story, I wanted to address one other question that we were interested in, which is the idea that microRNAs may act at a distance. And so one of the reasons that we pursued microRNA 425 is that it's actually expressed in great quantities in the heart, which makes sense, because that's where almost all of ANP and BNP is made. But microRNA 425, like many microRNAs, are expressed in other organs as well. And so it raises the possibility that other organs may use this microRNA to tell the heart to make less ANP or BNP. We know that many microRNAs travel in the circulation bound to HDL or carried in exosomes or vesicles. What this hypothesis was really speculation at this phase would require, of course, is that you can find microRNA 425 in the bloodstream. And in fact, Punkage demonstrated, if you look at the plasma of individuals, there are in fact quite robust quantities of microRNA that you can find in the plasma. This is using RT-PCR. And what's even more interesting is if you take individuals on a low-salt diet and measure their microRNA 425, and take the same individuals and transition them to a very high-salt diet, their circulating levels of microRNA 425 go down. And so this could then be a salt-responsive microRNA 425 microRNA, which then could allow organs like the kidneys and other salt-sensing organs to signal to the heart to make less or more of BNP or ANP. So just a couple other potential postulated causes of natriuretic peptide deficiency. Another one that we've been interested in is the idea that men in general have a relative natriuretic peptide deficiency compared to women. And the reason for postulating this is if you actually look at healthy men and compare them to healthy women who are otherwise alike, and these are data from Framingham, there's actually a striking difference between resting levels of ANP and BNP between these men and these women. In fact, the amount of variation in levels explained by gender alone is about 30%, which next to the genetic factors is the most significant factor for explaining variation in resting levels of both ANP and BNP. Carolyn Lamb was a postdoc with our group, did some studies, this is in Framingham, looking at whether this male-female difference varied by the hormonal status of the women. And so the men are shown here, these are log levels of BNP, and these are 4 groups of women according to whether they're premenopausal or postmenopausal, and according to whether or not they're on some sort of hormonal therapy. And as you can see, the biggest differences between men and women. Among the hormonal groups, although there are small differences, they're not really striking. And so what this suggests, since the hormonal groups are largely characterized by differences in estrogen, that this may in fact not be an estrogen-dependent finding, but may be something else. And so the something else is potentially testosterone. And so Carolyn looked now both within men and within women and looked at levels of circulating free testosterone and then levels of BNP. And what you find is within genders, there's an inverse relationship between circulating testosterone and BNP. And this is prominent in women, which is interesting because, of course, women don't have that much testosterone. But within the range of testosterone that you observe in women, women that tend to have more testosterone tend to have substantially lower levels of BNP, which supports the idea that this could be testosterone-driven. So to test this further, these are unpublished data in collaboration with Karen Miller and Joel Finkelstein at Mass General, Catherine Bachman, who's now with me at Vanderbilt. This is a study that Joel actually initially initiated in which 60 healthy men were given a GnRH agonist. As most of you probably remember from medical school, when you replace the pulsatility of GnRH with something that's non-pulsatile, it shuts down the gonadal steroid axis. And so given the men, this agonist basically shuts off their production of testosterone. These men were then randomized to getting replacement testosterone or placebo for 12 weeks. So functionally, at the end of 12 weeks, you have men who had restored levels of testosterone or men who had no testosterone, or sort of more physiologically similar to women in terms of testosterone. At the end of the 12 weeks, we measured levels of BNP and found that the men randomized to get replacement testosterone were, in fact, then had low levels of BNP compared to the men who did not have replacement testosterone. I know some of you were wondering who volunteers for studies like this, but anyways, is I think interesting and valuable data that support the hypothesis that this may be a testosterone effect, or largely a testosterone effect. The last thing, the second to last trait that I want to talk about associated with this handicap is race. And this is actually one of the nice things moving from working largely in the Framingham cohort to working with some other cohorts that we were able to look at this question. These work were done by Deepak Gupta, who's actually an alumnus of the medical school here, who's a faculty member with me now at Vanderbilt. These are data from the ERIC study. So ERIC has about 10,000 individuals, about a third black and 2/3 white, who had levels of BNP measured. And these are the cumulative probability distributions in blacks shown in the blue and white shown in the red, showing that the levels of BNP, the distribution of BNP is shifted leftward in African Americans. So African Americans tend to have lower levels of BNP than whites. These are the same data shown in a different way. If you just focus on the overall bar for one, this is a forest plot. This plots the relative difference in BNP between blacks and whites. And so if they had the same level, this dot would be on this dotted line over here at 0%. What you find is, on average, African Americans have 40% lower levels of BNP in the ERIC cohort. These other dots in the forest plot are then just looking at the subgroups. And so this is looking at people by whether they're less than 62 or over 62. And again, blacks compared to whites, whether they're young or older, have about 40% lower levels. And in fact, no matter what characteristic that you look at in all of these subgroups, there is about a 40% difference between blacks and whites in BNP. The other thing about ERIC that's nice is that we have access to their GWAS data. And so one thing you can do when you have access to these data is ask the question, for any given individual, what proportion of their genome is of European versus African ancestry? Because African Americans in this country are an admixed population. On average, African Americans have about 20% of their genetic background of European ancestry and about 80% from African ancestry, but there's a variation. And so you can plot this variation. So for each individual, you can look at their individual proportion of European ancestry and compare it to their BNP levels. And what Deepak was able to show is that as individuals increase the proportion of European ancestry in their genome, their levels of BNP are higher. Again, suggesting that the black-white differences are at least partly, if not largely, due to differences in genetic background, which we found to be a very interesting result. This has now been replicated in numerous cohorts. This is ERIC, shown here. Deepak has actually led all of this work. This is the Dallas Heart Study, MESA, the Cardiovascular Health Study, the Diabetes Prevention Program, and then the SD, which was a very large data set, was the synthetic derivative that I mentioned earlier from Vanderbilt. So these are now EMR-derived BNP levels. And really, in all of these cohorts, there's this consistent finding that African Americans have lower levels of BNP compared with whites. So very consistent. And so then, just to revisit where we are, what causes natriuretic peptide deficiency? Who has this handicap? I would postulate or submit that genetic factors, being male and being African American, are things that significantly predispose to having this natriuretic handicap, which in turn then promotes hypertension, LVH, obesity, diabetes. The last thing that I want to talk about related to this is obesity. Actually, one of the earliest things that we found in Framingham, shown here on the left, is that obese individuals tend to have lower levels of BNP than non-obese individuals. And this has now been shown in numerous cohorts. These are data from the Dallas Heart Study. This has been shown in healthy individuals, individuals with heart failure, individuals with cardiovascular disease. In almost every setting, there is a finding that obese individuals have lower levels of BNP levels than non-obese individuals. And this can't be explained by hemodynamics or other comorbid factors, which you can either adjust for, or in most cases, the hemodynamics are actually more unfavorable for obese people, so you'd think that they would have higher levels. And so this led to the idea that maybe there's also a primary deficiency in obese people, such that they can't make BNP or ANP quite as well as non-obese people. We've looked at this actually in mice. So this is in collaboration with Marielle Scherer-Crosby back in Boston, who looked at a bunch of standard C57 black 6 mice who were fed either a standard diet for a month or a Western diet, so a high-fat, high-fructose diet, and then sacrificed after 30 days. And in fact, they found that cardiac expression of the natriuretic peptides was substantially lower in the mice who were fed the Western diet, who were more obese after 30 days. Than the mice fed the standard diet. And I won't go through the further experimental data, but this was a finding that they found not only after 30 days, but actually, interestingly, even after short-term feeding with the Western diet, they were able to replicate lower expression of the natriuretic peptides. This actually motivated Pankaj, again, to do a different type of study, looking at whether there are acute changes in metabolic exposure that might lead to lower levels of ANP. And so for this, he took 30 individuals, either obese or lean individuals, and they were all standardized, again, on a standard salt diet before coming to the GCRC. And then once they got to the GCRC at time 0, they were fed a high carbohydrate challenge. It was basically a big glucose shake. And then they were kept in the GCRC, and their AMP levels and BNP levels were measured for the next 6 hours. And what he found is that independent of weight, so both the obese and the lean individuals had about a 25% reduction, even acutely, in circulating AMP levels after the metabolic challenge. Interestingly, this was more prominent, much more prominent for AMP than it was for BNP, which led Pankaj to hypothesize that maybe this was, again, another manifestation of the action of this microRNA 425. And so these are now cell experiments looking at cardiomyocytes exposed to glucose for 6 and 24 hours, showing that even after 6 hours, cardiomyocytes exposed to glucose have increased expression of this microRNA, this endogenous inhibitor of AMP, and concomitantly they have reduced reduced levels of glucose in the media after 6 and 24 hours. Pankaj went on to hypothesize that maybe this was modulated by a transcription factor, NF-kappa B, which is known to be induced by glucose. And so P50 and P65 are subunits of NF-kappa B. Overexpression of one or both subunits was associated again with increased expression of the microRNA 425. And finally, going back to these cell-based experiments, he found that again, if you expose these cardiomyocytes to glucose, they get an upregulation of expression of the microRNA 425. But if you then co-administer the glucose with an inhibitor of NF-kappa B, you can abrogate most of that increase in microRNA 425. Again, supporting the model shown here that glucose upregulates NF-kappa B, that in turn binds to the promoter and increases transcription of microRNA 425. The microRNA 425, again, is an inhibitor of AMP production, and so you find these cells exposed to glucose have decreased AMP production, which results in decreased circulating AMP levels, and lastly, the hypothesized short-term and long-term consequences of reduced circulating natriuretic peptides. And so the overall model now, at least to date, is that there are now a number of factors that we postulate are associated with a natriuretic peptide deficiency. Genes, which we talked about, testosterone, African American race, obesity, and then perhaps even acute exposure to glucose itself. And there are a number of ongoing studies now, not just that we're doing, but that others are doing, to investigate further the mechanisms in humans and animals, the obesity relationship, whether that's mediated through insulin resistance, and other factors associated with obesity. Overall, though, we hypothesize that having this natriuretic handicap would be expected to promote salt retention, hypertension, cardiac hypertrophy, and even an adverse metabolic profile. And so that leads to the final question of whether it's possible to reverse this natriuretic peptide deficiency. One thing that you might ask is, since obese people have lower levels, what happens when they lose weight? And so in humans, the easiest way to study this is to look before and after bariatric surgery, because that's the most effective medical intervention for weight loss. So Annabelle Chen-Tourneau, when she was a fellow at Mass General with our group, looked at about 130 individuals who were undergoing weight loss surgery at Mass General. And these were their BNP levels preoperatively. This was the average BNP at about 2 months post-op, and this was finally their achieved BNP about 6 months post-op. As you can see, there's actually a dramatic increase, more than a doubling shown on this scale, of BNP levels from before to after weight loss surgery. The dotted line shows the trajectory of BMI going in the opposite direction. So as they lose weight, their BNP levels go up. In fact, she also did echoes in these individuals, and over the same period of time, they had reductions in LV mass and lower blood pressure. So again, the increase in BNP levels was not driven by the traditional cardiac stimuli of increased blood pressure and LV mass, but in fact seemed to be a primary phenomenon of the weight loss. Similarly, we've looked at the chronic stage. So Jeff Walford is an endocrine faculty member at Mass General who looked in the diabetes prevention program, which consists, as you know, of several thousand individuals who are with prediabetes who are randomized to placebo, to metformin therapy, and to a lifestyle intervention of diet and exercise. At the end of 2 years, they had their BNP levels measured, and as shown here, the individuals in the lifestyle group had a significant increase in the NT-proBNP levels, which was not observed in either the placebo group or the metformin group. There is a TZD group shown here, which had an even bigger increase, but I don't show this data because of the potential confounding effect of salt retention. Of course, lifestyle and diet are difficult interventions to affect, and so the other question is whether there are pharmacologic therapies that might increase natriuretic peptide action in individuals with this handicap. And so there are a couple options that we have. One is direct agonism of the pathway, which is difficult chronically because it's a peptide, but it can be given subcutaneously, or there may be other ways with mimetics that it can be given. The second category are, of course, neprilysin inhibitors. These are drugs that inhibit neprilysin, which is an enzyme that breaks down the natriuretic peptides. And as many of you know, this is a component of the new drug from Novartis, LCZ696, which has now been approved as a heart failure drug. Thirdly, many anti-diabetic drugs, including GLP-1 agonists and DPP-4 inhibitors, likely augment natriuretic peptide signaling. GLP-1 agonists by directly stimulating natriuretic peptide release, DPP-4 inhibitors because they inhibit the cleavage of BNP into smaller peptides. And then lastly, PDE5 inhibitors, which I'll talk about in a moment. I just wanted to show as a proof of concept in animals that if you take animals and chronically infuse them with BNP, you get all the expected beneficial metabolic phenotypes that are predicted on the basis of the genetic models. And so you get, this is a glucose tolerance curve, you get lower levels of glucose in these mice, and this is the area under the GTT. You get improved glucose tolerance. And so you can recapitulate the genetic findings by administering BNP chronically, which again is a challenging thing to do in humans, which is why I show the mouse data here. But I want to close by talking about the PDE5 inhibitors. This again goes back to the cardiovascular pharmacology tradition at Vanderbilt that I mentioned earlier. Jackie Corbin is now an emeritus faculty member in the pharmacology group at Vanderbilt. Jackie spent his career studying cyclic GMP and discovered this enzyme below, phosphodiesterase 5, which was responsible for breaking down cyclic GMP. And in fact, Jackie hypothesized along with his colleagues that inhibiting this enzyme would result in increased availability of cyclic GMP and may have some beneficial effects. They were mainly interested in reduction of angina from vasodilation, but in fact, Jackie also postulated that there are other vascular beds in which cyclic GMP has vasodilatory effects, including the organs of sexual function, and had hypothesized that this may actually be a drug that would improve erectile dysfunction. And of course, that is where the story has really taken off with drugs like Viagra, shown here, or sildenafil, a PDE5 inhibitor for erectile dysfunction, but also happens to have vasodilating effects. Just as an incidental side note, neither Dr. Corbin nor Vanderbilt got any intellectual property from this finding, a result that led to a bunch of litigation, which was eventually turned down on appeal by the Supreme Court. But it's a good scientific story, at least. One of the current faculty members at Vanderbilt, David Wasserman, performed this study. So these are mice treated with sildenafil, PDE5 inhibitor, for 12 weeks. And David's group showed that these mice, at the end of 12 weeks, had less weight gain on a high-fat diet, those that are treated with sildenafil and L-arginine, a NO pathway stimulator. And at the end of 12 weeks also had improvement in their glucose tolerance. Again, supporting the idea that one of the beneficial effects of increased activity in this pathway would be the beneficial metabolic effects. This led to actually a number of groups, one Nancy Brown's group, and I don't show her data here, but also our group while I was back in Boston, to perform some pilot studies of looking at PDE5 inhibition and metabolic traits. This was done by Jennifer Ho, who's still at Mass General, and myself, looking at 50 individuals randomized to 20 milligrams of tadalafil, which is a relatively high dose. This is the pulmonary hypertension dose, versus placebo in a double-blind manner for 3 months. And at the end of 3 months, a number of oral indices, including this oral disposition index, were improved in the tadalafil group compared to the placebo group. ODI is basically a composite index of glucose, insulin, beta cell function, and insulin resistance. Data like these and data from Nancy's group led us to then initiate this larger randomized study, which has just begun randomization, which I am conducting with Evan Britton and colleagues, and that is also NIH funded, to look at long-term therapy with PDE5 inhibitors, in this case tadalafil, and a number of cardiometabolic outcomes, including energy homeostasis, measures of function in the cardiovascular system, and measures of function in the skeletal muscle. There will be a total of about 300 individuals randomized over the course of the next couple of years into this study. And so, just to summarize then, in returning to the questions that I posed earlier, we now believe, as well as I think many groups, that there are in fact states of natriuretic peptide deficiency which can promote increased cardiometabolic risk. The groups of individuals that may be particularly prone to this deficiency include obese individuals, men in general, and African Americans. And then lastly, it's possible, because this is a system through the natriuretic peptides and cyclic GMP that is quite responsive to pharmacologic intervention. This is a state that is potentially addressable through both lifestyle and pharmacologic therapies. So I wanted to, of course, acknowledge my many collaborators through the years that have contributed to this work, both at Vanderbilt, as shown here, and back in Boston at Mass General. This slide also shows our SFRN group for the AHA, my colleagues at the Framingham study, and at other institutions. I should probably specifically mention some of these collaborators. I mentioned Deepak, who's a junior faculty member working with me, who's doing many of the studies now. Back at MGH, Chris Newton-Che, a long-time colleague, Pankaj Arora, now at UAB, and the late Ken Block, who was really instrumental for many of the experimental observations that I showed here, and then again Vasan and Dan at Framingham. Many of you, I guess some of you have probably been to Vanderbilt, but I'll show these pictures. This is where we have our Clinical and Translational Research Center that was initiated when I joined Vanderbilt a couple years ago. This is Vanderbilt in the foreground, Nashville in the background, and of course the front of the medical center there. I greatly appreciate your attention. Thank you very much.
Well, thanks. That's a fantastic talk, really comprehensive. I'll start off with one question, the MIR 425 area. So sort of a three-part question. Where is it encoded, and what controls it? Sort of is an interesting spot. The second thing is, what are the other predicted downstream targets? And then the third, to ask you to speculate about the NF-kappa B regulation, given that that's sort of a blunt regulatory tool. I mean, almost all the bad stuff in cardiovascular is NF-kappa B. So is that sort of more of a broader, would you put a lot of other things on the left side of your diagram of regulators?
Yeah, those are great questions. So the MIR 425 is in a gene with not clear function, this DALRD3 gene, which really is a gene that has no known cardiovascular or metabolic significance. And so, you rightly point out that many microRNAs are in genes that make sense for the things that they regulate. This is not one of those cases. And so
most of them are.
Yeah.
Most of them are hidden in funny places.
Yeah, and so we don't know whether, it may just be there and regulated by distant factors. We're really quite early in that knowledge. The second question having, certainly the NF-kappa B, as you point out, is a ubiquitous transcription factor, and we've not looked at other things that might influence the MIR and ANP either through NF-kappa B or whether there are other transcription factors that might be responsible for this finding. Yeah, it's a good question, though.
And then other targets of 425?
Oh, yeah. So Pankaj just looked. If you look bioinformatically, one potential downside of bioinformatic looks is you tend to see what you want to see. And so if you look bioinformatically, there are in fact many genes that could be bound by MicroRNA 425 that are in the metabolic pathway. like PGC1 alpha, and there are other genes that look like they potentially could be related. So there could be the makings of a overall metabolic regulator story, of which the natriuretic peptides are just one arm of it. But I would hesitate to be to make too strong of an assertion about that, because it's a good number of genes and they have many functions.
Other questions from the group? Peter? You gotta turn it on. Hold it down for a second. Yeah, I'll turn it on.
So you mentioned the DPP4 and the others are the GLP-1s, but there's a new molecule on the block, the SGLT2s, which work at the kidney level.
Yep.
And for blocking sodium reabsorption and probably other analytes. So the question is there any data on these different molecules right on the cardiac function itself? Because, for instance, with the empagliflozin trial, it's been hypothesized that there may be natriuresis and glycosuria, and it's acting as a long-term diuretic, but then it appears to protect against heart failure.
Right.
Is it working directly? And it takes people like you to do these experiments directly on the heart, these newer drugs, which we think are working on the kidney, but are they working on the heart too, directly?
No, of course, that's a great question, and I don't know the answer. I don't know that it's been looked at. I've seen, some data suggesting some associations with intrinsic cardiac phenotypes, but I don't really know, and I don't believe there have been any direct studies done with these inhibitors. Obviously, they're quite new and still being investigated in the phase 3 outcome studies for cardiovascular disease. I concur. These are questions that need to be asked. I think these are new agents, and so it would require some collaboration with industry to help facilitate these types of studies. There are more data, of course, with GLP-1 and DPP-4. Again, I think it's complicated, and I didn't get into it much. To attribute the potential cardiac beneficial effects of any of these drugs to this one pathway would be, of course, an overreach, because all of these drugs are so pleiotropic. And so DPP-4, although it does cleave BNP and AMP, or BNP cleaves many other peptides. And it's even true of the cardiac drugs like LCZ696, although many people assume that the beneficial effect in heart failure is largely driven by the natriuretic peptides, because of course it's logical. There are obviously 20 or 30 substrates of neprilysin, and so we can't even be sure in that case, which I would say is an even more obvious case that the natriuretic peptide system is playing a big role. But we don't even have definitive data in that case that's going on.
Excellent talk. Trying to take it to a clinical level, there's a strong contingent of cardiologists that are pushing biomarker therapy of heart failure and biomarker diagnosis of heart failure. The problem I've had with it all along has been the wide variability in the values that you get, which you've did an excellent job of explaining the reasons for that. But can you give us any guidance as how to use these clinically?
Yeah, it's a good question. I think it is true there's wide variability, although I think people who would support the use of biomarkers like BNP would argue that's true of almost anything. I mean, if you look at LDL, the intra-individual variability in LDL is quite large too, but obviously it helps us target therapy. I would characterize the prospective data on the use of BNP for guiding heart failure data as modestly supportive, and with the emphasis on modest. There, of course, been a number of both positive and negative studies. The NIH is currently supporting a relatively large study in that area called GUIDED. There was recently a study reported at HFSA just a couple weeks ago, which was negative. So, but we use it. We partly use it because you can look within an individual, and so I would say comparing different individuals and their levels is particularly dangerous because of many of the factors that I mentioned. But many of the factors that I mentioned do not change over short periods of time, and so if you look within an individual and see dramatic increases or decreases in the peptides, then you might safely assume that there's something going on related to their heart failure. Of course, even then, as I showed, if they happen to have eaten a Big Mac before coming in, then that might cause an acute change in their levels. So even in that setting, I would take it with a grain of salt, no pun intended. I think the other point to make is that although our first line of thinking about biomarkers is that they use that they allow us to risk stratify, which I think is, I mean, I do the studies like that. Many in this room have done studies like that. I think the real promise of biomarkers is to help us choose the right therapies to give, the therapies that are suited to the pathophysiology. So again, going to LDL, I think the reason that LDL is a good biomarker is not so much that it's such a great predictor of risk, and Peter may or may not quarrel with this, but because it suggests what to do. Because someone with a very high LDL, there's a particular etiologic pathway that you can go after. And so I think ultimately our best biomarkers are the ones that help us decide what therapy to treat with, as opposed to just that the patient needs therapy. And so again, going back to the BMP story, I would say that and this is, thinking in the clouds right now, if one were to come up with an endocrine assay for figuring out who really does have this deficiency in natriuretic peptides, then it would suggest that those people are potentially the people who you'd most likely treat with a therapy that either increases natriuretic peptide production or augments the pathway in some other way. Just the same way that we don't give everyone thyroid hormone. We give people thyroid hormone who have evidence of being deficient in that pathway. And so, again, getting to the BNP thing, BNP as a therapy for heart failure, acute BNP, if you think about it, you're giving IV BNP to someone who's already making a lot of their own BNP. And you can argue about whether that BNP is functional or not, but I would make the argument that the people who would really stand to benefit from BNP-like therapies are the ones who have intrinsic deficiency in their ability to make BNP.
Let me just ask one more. You talked about the differences in African American, Caucasian populations and the polymorphism. What about other populations, Asian, and perhaps even more interestingly, the Latino population with their cardiometabolic differences?
Yeah, also good question. So we don't have a lot of Asian data. We've looked in MESA, and there's some slight differences, but the numbers aren't so big. We have looked in Hispanics, and there looks like Hispanics do have a somewhat lower level, but not nearly to the degree that African Americans have. And that's actually interesting to us because Hispanics are also a little bit admixed as well. And even in that population, there is evidence that there is tracking in a similar way to the African Americans, that it does track with ancestry. And so, but there are data that currently we're working on through MESA and some other cohorts, but trying to acquire more data.
Well, I'll ask you all to join me again. Thank Dr. Wang for a fantastic talk.
The preceding program is copyrighted by Emory University.